afternoon. My name is Susan Howlett, and I'm going to be the moderator for this exciting session on cardiovascular disease and sex differences in its presentation. I'm a professor of pharmacology and geriatric medicine at Dalhousie University, and I'm going to be interviewing Dr. Kararigas. Introduce yourself.
Sure. My name is George Kararigas. I'm a professor at the Faculty of Medicine of the University of Iceland, and I'm very happy and excited to be here with you, Susan. Thank you so much.
So maybe you'd like to start by telling us a little bit about your study, and maybe even first to talk about what do we understand when we think about sex and gender differences in cardiovascular disease?
Sure, that's a very important question because in the field we use sex and gender quite often in not always an appropriate manner. So when we talk about sex, we are considering biological aspects such as sex chromosomes and sex hormones. However, when we discuss gender, then we're taking into consideration things that have to do with socio-cultural aspects. So gender is actually a socio-cultural construct that we have come up, and in contrast to biological sex, as our society evolves and progresses, gender has many different aspects, and people identify with different types of gender now. So we live in a spectrum in regards to gender nowadays.
And perhaps you could tell us a little bit about the experimental work that you've presented today.
Sure. My presentation mostly was on biological sex, actually, not so much on gender. And we started looking at patients with pressure overload. So these are patients that have aortic valve stenosis and they undergo aortic valve replacement surgery. And since we try to understand the differences that we see between male and female patients in terms of their clinical manifestations, we have harvested some tissue samples and performed omic analysis so that we can identify the underlying mechanism. We were very excited to see that the transcriptomic regulation of those cardiac tissues differs significantly between the male and female hearts. We saw that the male hearts have pathways that are related to fibrosis being induced, so upregulated, while the female hearts, we saw that pathways related to inflammation, for example, they were repressed. And we consider this something like a cardioprotective mechanism that is active in women.
That's very interesting. So that's a clear example of why it's so critically important to include both males and females in studies. What about with respect to preclinical work in experimental models?
Yes, I mean, it's very important because we frequently see that a lot of our new targets, they don't make it into clinical trial phases, or if they do, then they fail, they don't live up to the expectations. And one of the main reasons, I should say, is that in our preclinical research, we don't, there are many of studies that do not consider sex as a biological variable, basically. And they use either male only or female animals, in most cases male animals, I should say. They hardly know the sex of their cell lines when they do in vitro work, for example. So it's mostly often that what has been found and seemed to work in one sex, this does not mean that it will be the same in the other sex.
So you've identified sex differences, and I wonder if you've started to look into some of the underlying mechanisms involving sex-steroid hormones that might regulate these.
Yes, that's a very interesting and important question. We are looking primarily at estradiol, what is considered to be the female sex hormone. However, I would like to mention at this point that both men and women produce estradiol locally. And this takes place via aromatase that converts testosterone to estradiol. And particularly there are some situations like obesity due to the fact that the adipose tissue is a major contributor of local production of estradiol, where men might have significantly higher levels of estradiol than women even. So with this in mind, we have taken a number of approaches to try to understand the role of estrogen and estrogen signaling in the manifestation of sex differences in cardiovascular diseases. And we have seen that, for example, in the absence of estrogen receptor beta, which is one of the main estrogen receptors, the response to pressure overload differs significantly between the male and female mice that we used in the study.
That's fascinating. Do you have a take-home message from all of this work that you'd like to talk about right now?
Sure,
Yeah.
sure. We are currently in the era, still, I should say, of the one drug, one dose fits all approach. So if we want to move a step closer towards personalized medicine, we need to consider both sex and gender so that this can bring us into a more tailored medical care for both male and female patients.
Well, thank you very much. And this was a great talk in a session full of great talks, and it was a really interesting session on sex differences in cardiovascular disease. It's an emerging area, and it's critically important for us to do research and include both males and females in both clinical and preclinical work. Thank you.